The main focus of our research here at VeroScience is understanding how the biological clock system within the brain regulates metabolism throughout the body. Over the past 50 years, there's been an explosion of obesity and type 2 diabetes across the globe that simply cannot be explained as a function of a genetic defect. We have been studying animals in the wild under natural conditions that exhibit the natural onset of the obese insulin-resistant condition at particular times of the year as a survival mechanism against low food availability. And the obese insulin-resistant condition allows for increased hepatic glucose production to keep the brain functioning when none is available in the environment. This annual cycle of metabolism, moving from the obese insulin-resistant to the lean insulin-sensitive condition, or from the pre-diabetic to non-diabetic condition, is the result of changes in circadian patterns of neural activity at the clock center in the brain. At one particular time of year, there is a particular 24-hour pattern of neural activity that induces the obese insulin-resistant condition throughout the body via the clock's communication via the neuroendocrine axis. That circadian pattern is shifted in the springtime to induce a different signal to the clock to induce the insulin-sensitive condition. Over several decades of research, we found that a particularly important neurotransmitter circadian rhythm that regulates that metabolism message to the clock is the circadian rhythm of dopamine. In animals that are obese and insulin resistant in the winter, the circadian peak in dopamine activity is dramatically diminished, allowing for the expression of the obese insulin resistant condition by the brain. In the spring, it's the opposite. That circadian peak of dopamine activity is very high, and that sends a different signal to the brain that programs it to modulate metabolism throughout the body to the insulin-sensitive non-diabetic condition. What we have found that just as the winter condition is characterized by a diminution of the circadian peak activity of dopamine at the clock, the westernized lifestyle induces the same situation. High-fat diet, sleep-wake cycle alteration, social stress, altered photoperiod, all function to lower brain dopamine activity, particularly at the biological clock, sending the message to the brain to program the body for the obese insulin-resistant condition. So it's more a matter of the brain's taking over control of what you do with the calories you take in, rather than the actual number of calories you take in. So the hope is we'll be able to provide nutrients or pharmaceuticals at the appropriate times of day that reset the clock and maintain the clock in its normal cyclic pattern at the appropriate times of year, just like we see happening in animals in the wild.
We're interested in the effects of high-fat diet on fuel sensing by the brain. And to address this question, we introduced electrodes into the ventromedial hypothalamus of anesthetized rats and recorded multi-unit activity from a small population of neurons around a recording site, while locally injecting 5 millimolar glucose to those cells. This is an example of a recording we obtained from an animal raised on a regular diet. This first minute here to the left shows spontaneous activity in response to endogenous glucose. We then presented a 1 minute 5 millimolar pressure injection of glucose to the recording site, and we see this this strong elevation in firing rate indicative of appropriate glucose sensing. We then took animals and raised them on a high-fat diet and repeated the same experiment. And when we do this experiment, we see a much diminished response to the 5 millimolar glucose. Now we know that low brain dopamine is associated with an insulin-resistant state. So we then took these same animals that were raised on a high-fat diet and gave them bromocriptine, a dopamine agonist, at the change of their light cycle, right as they're waking, and did the same experiment. And we see a robust response to the 5 millimolar glucose injection, very comparable to the type of response we observed in the animals raised on a regular diet, thus showing a restoration of appropriate glucose sensing in the hypothalamus.
Type 2 diabetes is a major problem across the globe, and it carries serious morbidity and mortality risks, increased risk for cardiovascular disease, shortened lifespan, and compromised quality of life. But by being able to understand how the body naturally regulates metabolism and mimic the natural mechanisms for regulating metabolism, we hope to reduce the incidence of obesity, type 2 diabetes, and their comorbidities.